Good afternoon, everyone. My name is Jen, and today I'm presenting my MSc Bioinformatics project titled Transcriptomic Profiling of Daphnia magna across polluted river sites. This study integrates environmental chemistry, transcriptomics, and statistical analysis to explore how pollution impacts gene expression in a key aquatic indicator species. The aim is to uncover biological responses to chemical stressors and assess how location influences transcriptomic variation. This project was part of a broader ecotoxicological study investigating polar organic chemical contaminants in a river system flowing through multiple cities. Water samples were collected from 12 geographic sites, ranging from upstream D12 to downstream D01. We used Daphnia magna neonates in the bioassays to assess the biological impact of these pollutants. The aims were to explore how the polar organic compounds affect gene expression in Daphnia magna, use transcriptomic data to identify location-specific biological responses, apply statistical and machine learning approaches to uncover predictive patterns. This research contributes to understanding how pollution affects aquatic systems, ecosystems, and potentially human health through waterborne exposure. Each water sample was concentrated between 100,000 and 250,000 times in environmental level. These extracts were split for chemical screening and bioassays. Biochemical analysis covered 91 compounds, while the bioassay exposed Daphnia magna neonates to the extracts. RNA-seq was performed on the exposed samples to capture transcriptomic changes. This design allowed us to correlate gene expression with geographic pollution gradients. We merged 3 data sets: the sample sheet, RNA-seq expression matrix, and gene annotations. Each row in our final data set represents a sample and each column a gene. We retained the location column for grouping and focused on numeric gene expression values for statistical analysis. This structure enabled site-level comparisons and downstream enrichment analysis. To identify biologically relevant genes, we binned them by mean expression. Genes with expression above 6 were considered highly active. This gave us 11,086 highly activated genes, of which 5,226 were fully annotated. This filtering helped us focus on genes with meaningful expression levels across sites. Visualizing these bins revealed a skew toward moderate expression with fewer genes in the highest bins. We performed principal component analysis, PCA, to visualize sample clustering. Samples from CK sites clustered tightly, suggesting consistent expression patterns, and this is the control group. In contrast, samples from D sites were more dispersed, indicating greater heterogeneity. The PCA plot showed that PC1 captured the largest source of variation, reinforcing that location is a major driver of transcriptomic differences. Cumulative variance analysis showed that the first few components captured over 90% of the variance, justifying dimensionality reduction for the downstream model. We ran one-way ANOVA across all sites to identify genes with significant expression differences. Out of thousands of tested genes, 168 showed statistically significant variation, p less than 0.05. This suggests that pollution levels or site-specific factors are influencing gene regulation in Daphnia magna. However, when we ran Tukey's HSD post-hoc test, no pairwise comparisons between sites were statistically significant. This may be due to high within-group variability or limited sample sizes. Nonetheless, the overall ANOVA signal supports location-dependent transcriptomic shifts. We use GeoProfiler to analyze the top differentially expressed genes. Enriched biological processes include cell proliferation, Geo 00082883, positive regulation of cell cycle, Geo 004787, G1S and G2M transitions of the mitotic cell cycle, and cell cycle arrest. These results suggest that exposure to pollutants may be triggering stress responses and disrupting normal cell cycle regulation in Daphnia magna. This aligns with known toxicological effects of organic pollutants on aquatic organisms. We trained a classifier to predict sample site based on gene expression. Initial results showed poor precision for some sites due to class imbalance and limited sample representation. We chose not to use SMOTE for balancing, as it would have required dropping underrepresented classes from this top 20. Feature importance analysis highlighted key genes driving site prediction, including DAPMA 7B EV M011091, which was highly expressed in DO5 samples. This gene may serve as a biomarker for pollution exposure and warrants further investigations. In summary, our analysis shows that environmental pollution along the river correlates with transcriptomic changes in Daphnia magna. We identified 168 significantly differentially expressed genes and enriched geoterms related to cell cycle regulation. Although pairwise site differences weren't statistically significant, the overall patterns suggest biological impact. This research highlights the utility of transcriptomics in environmental monitoring and the potential for early detection of ecological stress. Given that Daphnia magna is a model for aquatic toxicity, these findings may also inform human health risk assessments related to waterborne pollutants. Future work will focus on integrating chemical concentration data, improving classifier performance, and validating key pathways through functional assays. Thank you, and here you can find a list of my references. Goodbye.